So I think we'll just start the session. Um, so I'll be sharing my screen. Uh, Please let me know whether the slides are clearly visible or not, and whether I'm audible. Okay. Thanks a lot. Yeah. So again, I welcome all of you to week 12's content for the course Bioinformatics Algorithms and Applications. I am Sunanda Samant, PhD scholar from IT Indore, and I'll be uh, taking the live tutorial session for today's course. So this is essentially kind of the main final week's content from the NPTEL course instructed by Professor Gomiha. So of course, as you might have already gotten an email from, that there might be some additional sessions that the TAs decide to do, like let's say one extra day for 2 hours or 1 hour class basically. So I have already communicated with the NPTEL team that I'll be doing one basically extra kind of live tutorial session, kind of an additional class. So that is tomorrow from 6 to 8. So I think you should have probably received the email for as well as the timing and the links also. So yeah, so since this is the kind of the main final week's classes, so we'll discuss some of the questions uh, and I guess some of the concepts that that were in the week's content, and also uh, kind of the questions similar to what came in the earlier iteration of the courses in the assignment portions. But also, so that will be like quite a brief set of questions, and since that is kind of the end for the entire coverage there. So what I will do is that I will start kind of recapping everything that I have gone through from, let's say, week 1 right up till week 12, right? So a few of the recaps I will just cover in this class, and the rest of the ones, basically the remaining ones, I will talk about in tomorrow's 6 to 8 class. So kind of just a recap class to wrap everything up, right? So that is the plan for today and for tomorrow's session. So yeah, so let us quickly begin, I guess. So in the previous classes, we went over some of the concepts regarding the some of the concepts regarding the validation of algorithms and such. For example, we learned about receiver operator characteristics uh, and the different terms that kind of indicate how a machine or algorithm is kind of performing, right? The evaluation sort of metric. So from that we learned about these different parameters such as the true positive rate, false positive rate, and so on, which can be calculated by just counting how many true positives, true negatives, false positive, and false negatives are we getting from the prediction by the algorithm, right? We talked about how we can evaluate performance through the ROC curve. For example, we here talked about a true positive equals false positive rate. Then that is kind of a random prediction. Then we also learned about the different sort of metric that are commonly used. For example, sensitivity, which is again true positive rate, specificity, which is true negative rate, and then the false positive rate can be indicated by 1 minus specificity, and so on. And then accuracy can be the correctly predicted values divided by all of the positives and negatives that are present in the data, basically, right? Then we learned a little bit about awk programming, which is basically a very powerful tool to parse through text and a lot of data files, since a lot of bioinformatics uh, essentially analysis or data sets, or even if we talk about computational kind of data sets, for example, in MD simulation trajectories and so on. So when we analyze those things, generally the output files are stored in something sort of a text kind of file, right? Maybe a .txt, .dat, or something like that, right? So those are text editable files, of course, and you can parse through those and retrieve, let's say, useful information regarding those through something like awk command, basically the awk programming or the awk command lines, right? And from that, we just learned about a few brief like syntaxes, kind of, right? For example, the dollar signs are basically indicating columns and such, right? So from a data set, if we have like a let's say a data.dat file or something like that, right? So you can use the awk programming to basically paste on a terminal different things about the data files, such things, right? So that is how it can be useful while parsing the basically the data files or the text editable files essentially, right? So we learned a few things about those. Then we solved some examples regarding how to calculate the sensitivity, specificity, and so on, right? So from experimental and predicted values, you have to.
Excuse me,
Yeah,
can I
please.
ask something?
Sure. Please.
In the previous awk thing, so I downloaded because I'm new to this thing. So I attended Ramakrishna's session also.
Okay.
So I downloaded awk,
Mhm.
and he shared some demo files.
Okay.
So, but I'm not able to run awk. Maybe, matlab, I'm sure I'm doing some mistake. So I'm using Windows, and I have downloaded Mobixterm, which is a WSL, Windows Subsystem Linux System.
Yeah.
So there, so matlab, I'm not able to go to the place where I downloaded the files or saved my files. I need to be in that directory, right?
So
To
I think wherever you have installed the Windows subsystem for Linux, I think probably you have to go there and then try that, like open the terminal from there, something like that, and then you can maybe try running the commands. So like, how do you basically start the Windows subsystem? So you open up your command prompt or PowerShell, something like that, and then
no, I'm just from the start menu. I just start my mobile stump,
okay, and after that,
and
does it open some kind of terminal?
that is working like a Linux system only.
Okay, yeah. Then you should be able to open a terminal and then type the awk commands there. No.
But I have to reach that file now where that Ramakrishna has shared with us, or
Okay.
that is just an example. Or if I have some other file which I need to manipulate,
Yes.
so I need to go there. I have to specify, like here in the
Yes.
in this page also you have mentioned in the last data.date. So
So yeah.
so supposedly my file name is also data.dat.
Yes.
So I have to be in that directory now where
Yes.
my file is.
Exactly. So you have to be in that folder or in that directory where the data.dat is located. Otherwise, what you can do, let's say if you are like your data.file file is in some other folder. So what you can also do is that you can copy the path to that particular folder, and then you can use, for example, so let's say your data.dat file, you are currently in a subfolder, and your data.dat file is in a folder previous to this subfolder. Like you're currently under a subfolder of the main folder where the data.dat file is there. So you can probably put the path in something like this. For example, // indicates that I am going back a directory.
One folder up.
Yeah, one folder backwards. So
okay.
yeah. So for example, if your data.dat file was 2 folder backwards, and then let's say it was in something like test data folder, inside these 2 folder backwards. Like if you go back 2 folders and then there is another folder called test data. Within that you have data.dat file. So then you have to give the proper path for that data.dat file by something like this, basically. So this is in relation to where you are starting your terminal, basically in which folder you are running this awk command from. So if you are running from the exact same folder where the data.dat file is located, so you do not need to give any additional path. But if it is in some other directory, then you have to give proper path for that directory. Yeah.
And how to open this awk application? Like I have downloaded it is somewhere in my C drive and in programs folder, and then inside, and there was one folder bin, and inside that there was this awk and gawk, and 2-3 applications down. They downloaded.
Okay.
So after that, what am I supposed to do?
So it should run from the terminal itself, right? If you open up your Windows subsystem for Linux, and then if you just type in awk, does it work?
I typed which awk, so it was working. And in some like file which I created there itself, echo and some file, and from there I exist and I try to run the file $1 plus $3. So it gave me, after the printing, it gave me the answer.
Yeah, that should be okay, right? So If it is installed
So that
already,
means
then it
awk
is,
is working.
yeah, yeah,
Awk is working.
awk is
But
working, awk is working.
so matlab, now I just now I mentioned na, I'm not able to get to my this data file,
Hmm.
work with my data file, so that is the issue. So now you just give me a clue. So from my awk, it is somewhere in my C drive, then programs folder, and inside one or two folders, and
Yeah,
inside bin it is awk and gawk. Which one should am I supposed to open? And I double clicked that, but it didn't open, it just
no,
opened and closed.
yeah, yeah. So I think you should
So
try running this from the terminal itself. So for example, okay, let me just do one thing. Just a moment. Uh,
so from Windows I can use PowerShell, right?
yes, from Windows you can use PowerShell. I actually haven't downloaded any Windows subsystems, but so let's just say something like that. Yeah. So let's say you are like currently in a directory where you have all of these files, different files. For example, this rmst dot that something like that, right? You can probably see via some text. That is, there is nothing there. Uh, yeah. Let's say you have some file like this, right? Now, if you are inside this particular directory, okay,
Where
yeah,
my data file is
where
there.
your data file is, then you can run off directly from this one. Okay. Sorry. Let me do one thing. Let me just share from this one only. Okay. Yeah. Is it visible now? Yeah, I think it should be. Yeah. So let's say you are trying to just uh, print, let's say the lines from this one, right? So this RMST underscore back underscore new dot dat. Then you can, as usual, basically uh, keep the command from here /t. Then /0. Yeah. See. So this is basically your rmst underscore back dot dat, right? Now, on the left hand side column, you will see if you go to the very end of this file. So what you will see is that the left hand side column is kind of indicating what is your line in this data file. So if you will notice at the bottom right corner, so it is mentioning the basically the line number, like where you are currently at, right? And then the middle one is basically telling you about your frame number. So this is from a molecular trajectory data. So this will tell you how, at what kind of frame number or at what snapshot number you are currently in. So if you go to the exact end, you will notice that the first basically column in this data file, it is showing 20,001. So that means that the total number of line that you are currently at is 20,001, and it is 20,000th frame, and it is showing you the corresponding RMST value for that. Because in the first line you see a hashtag frame and some other RMST underscore 0006 was written there. So that is not part of the RMST data. So that is why at the very end you are seeing it is 20,000 only. So -1 from the total line number kind of. So when you run this basically awk command, so what it does is that it prints out the number of line uh, basically from in this portion particularly, you will see. So this $0 is kind
So
of indicating, yeah, yeah, yeah.
$0 means the line number.
$0, yes. $0 means the column number basically, not the line number.
Oh,
Line
sorry,
number,
column
yes.
number.
Yeah, yeah, yeah,
Mm.
yeah. So in that way, you can kind of parse through that. So let's say we try another one here. Uh, let's say, so in this one, what it says is that it is basically printing you the number of lines. So basically the end part
Mm.
of the data. So total number of lines are kind of printed here. So that is 20,001 in this case. So like that you have to do. Now let's say if you had, let's say I'm just randomly making some new folders. So let's say I am making test here, and then I go inside test. Let's say I am making another folder inside test, which is called test two. So I am now inside test two. Okay. Now inside this test two folder, I do not have any data file that I want to basically parse through or read, right? So what we can do, since our RMST back.dat or back new.dat, that was kind of two folders backwards.
Two folder.
So
So
yeah, we
dot
can,
dot slash.
yes. So we can here, if you give this RMST back new.dat, it will not work because it will say that there is no such file or directory. But you can give the proper path through dot dot slash backwards. That means one folder backwards, and then another dot dot slash means two folder backwards. So now if you write that, now you'll get the result. So you have to give the path for the data file that you are trying to basically parse through. Uh,
Or else you have to be in that where the data is.
uh, yeah, I mean, could you please say it again?
Either I have to be in the folder where my data is present, or
Yes,
the other way around is I can give the path, the complete path.
yes, yes. Either you have to be in the folder where your data is present, so you can directly write the name of the data file from that, or otherwise, if you are trying to run off from another terminal, so you have to give the proper path for that particular file, basically. So another way I could have done is that, let's say I would have just printed the actual path in my system. So rather than going dot dot back, dot dot slash and dot dot slash, so two folder backwards, I could have just printed the entire path for the data file, basically where it is staying in my system. I could have copied that, and instead of basically this dot dot slash, I could have given the entire path. So in my file, it is in this home Kurt3 project Sunando, something like that. So in this folder, this file is staying. So I could have done that also. So in your Windows, it will probably be like in a drive C, then
C
forward slash.
drive
Yeah,
and then.
yeah, yeah.
Okay.
That way you have to
Okay.
copy the path, the entire
Okay.
path basically. Yes.
And this awk I can operate from PowerShell also, right?
I think you should be able to operate from PowerShell
I'll
because
I'll try. I'll try.
yeah, yeah,
Thank you so much.
yeah, yeah. Okay, okay. Um, yeah. So I'll just share the screen on the other system. Yeah, so we learned about the true negative, true positive, false negative, false positive, those kind of parameters, and how you can compute the or calculate the evaluation metrics from those set of parameters, right? So that this can allow us basically to calculate sensitivity, specificity, and so on. Uh, and yeah, we like talk for one question about what neural network is. Basically, you have an input layer, one hidden layer, and then another output layer. So that is kind of what we talked about. Then we solved one problem where the confusion matrix was given. So it is basically the prediction of an algorithm in a matrix form where the actual value versus predicted value is given, and you can compute again the general sets of metrics from that, right? So these were the kind of questions and basically uh, the algorithm evaluation parameters and such things that we went over in the earlier class. So in this class, we'll just take a talks about some concepts in continuation to that, and then, as I said, after that we'll just start our recap for what we did. Starting basically from week one itself, right? So yeah, so one of the databases, basically what Professor Gomiha went over was PDBTM, right? So there was one question in one of the assignments for some of the earlier iterations that this PDB-TM, what kind of data basically it does provide to. So we all know about PDB, the protein data bank, right? So it holds basically the deposited 3D coordinates of crystallized biomolecules, right? For example, through X-ray diffraction or through cryo-EM studies, if some research groups have uh, basically crystallized some protein, let's say in complex with some ligand, or some DNA in complex with some protein, something like that. So they will submit it or deposit it to the PDB database, and we can access it from there, right? So what does PDB TM provide the data for? So PDB TM, basically what does TM stand for in this PDB TM term? Yeah, I think on hand is there. Yeah, please.
Transmembrane, so it will be D, I think.
Yes. So the PDB-TM database, it basically has the structure as well as the sequence information for the transmembrane proteins, so transmembrane helical proteins or strand proteins and such. So this could be again another useful database what you can refer to if you need this kind of particular data for some project or research, right? So that was one database. Now let's say since we are talking about algorithms and the evaluation and predictions and such things, right? So if you have to predict protein stability, again it is talking about some real value for the protein stability. So what kind of analysis would you say that we can do? Can we do principal component analysis, KMS clustering, or seek to feature, or linear regression? So what kind of analysis can be performed, or what kind of algorithm can be used uh, to predict protein stability? Again, this could become a little bit confusing as well, the particular answers, but yeah, I see one answer B. So in this case, you can use a linear regression. So what are these different kind of algorithms? So principal component analysis is basically, so if you have a higher dimensional data, for example, when you talk about molecular dynamics trajectory, it's a 6n dimensional data, right? There are a lot of atoms, so n number of atoms are in this system. So if you imagine, let's say a system is consisting of 80,000 total atoms, right, including water and everything. So for that, if we just directly want to infer some useful information, it can be quite challenging. So what this principal component analysis does in that sense, what we can do is that we can take some of the 3D coordinate as input, and we can basically uh, perform this analysis in a way where ultimately we'll get the principal components, which will be in order of the most significant to the least significant. Okay. So in that way, you can do principal component analysis where uh, basically you are basically projecting the eigenvectors and the eigenvector projection for on the principal component one. So that will give you the most significant information about the data, or the most significant variation will be captured by this particular uh, principal component, the first one. And after that, if you talk about principal component two, then the contribution for capturing the variance in the data for that particular principal component will be even lower than principal component one. And then if you go down the path, so all of the principal components that you want to calculate, you will quickly notice that their significance decreases by a lot, right? So you can basically from the n-dimensional, from the 6n dimensional data from the memory trajectory, you have now gotten ultimately, let's say, a total of 10 principal components that can capture the variance in the data with, let's say, 80% accuracy, something like that, right? And the first two principal components, or the first three principal components, let's say, they capture the total of 50% of the variance in the data. So it can be basically a way to quickly uh, get useful information from a data where the data itself is higher, basically dimensional, and it is not always quite possible to, in a straightforward way, to extract any meaningful information from there. Then another one algorithm that it is talking about here, it is k-means clustering. So k-means clustering, as we know, that clustering is basically meaning grouping, right? So if you have an entire data set, you want to cluster it. That means you want to group this particular data set into some sort of groups where within each group your individual some sort of data points will be clumped at, right? So k-means clustering is another algorithm where it basically, we talked about in one of our earlier classes, where basically it chooses based on how many clusters you want to calculate. So it chooses a random point in your data set as representative of that cluster, and then from that it determines, okay, which other points can be a part of that cluster, kind of in a distance kind of sense, right? uh, and then after that it calculates kind of an average of these data points from that, uh, and then in the next basically after that calculating the average, what it does is that from each data point it calculates the deviation, and then you have a total kind of deviation from this data point that you are getting, from this data set that you are getting, right? And in the next iteration, what it will do is it will start again with some other random points, and then it will try to basically allocate all of the other remaining data points into particular cluster representatives, and then it will calculate the basically within cluster sum of deviations, right, sum of variation. And then ultimately when following many iterations, when it cannot improve this deviation value from the previous iteration, then the algorithm will have been converged, and then you will get your distribution of the data points into particular sets of clusters, the number of clusters that you want to have. And we talked about that WCSS, or within cluster sum of squares, is one of the useful kind of ways to know how many clusters you can divide your data optimally into. So there is one elbow plot. When you look at the elbow, so it can basically give you the optimal number of clusters, because after that, if you increase the number of clusters, the significance of that is kind of not that much, right? Seek to feature is, of course, you are giving a sequence, and in turn, it is kind of predicting some sort of features, right, from that sequence. So that is an algorithm for that. And then linear regression is basically where you have like a dependent variable, which can be expressed in terms of different independent variables. And ultimately, this dependent variable is kind of what you are trying to predict, right? So in this particular sense, the prediction of protein stability, so that will be your dependent variable for the linear regression. And then you can express this in terms of different other parameters, let's say, right? So protein stability could be in terms of your long range contacts, let's say long range order, something like that. So that could be one parameter. To the number of helices, number of sheets that is within this 3D fold of the protein, that could be some other parameters, right? So those could be the dependent variables, and the independent variables will be the combination of kind of those dependent variables, right? Let's say the number of loops, or the total number of residues that are part of a unstructured loop region or a coiled region, right? So that could be another dependent variable. So from all of these different sort of sets of variables, ultimately you're trying to predict what could be the stability of this protein. So you are trying to make an equation uh, from basically using all of these different parameters, ultimately trying to predict your actual kind of target, right? Independent variable, that thing. So in this case, the question was kind of asked in that way, because protein stability can be represented in, let's say, different sort of structural terms or parameters, such as I said, it could be like long range order, it could be number of helices, number of sheets, it could be the total percentage of coil structures or unstructured loop regions present in the protein, such things, right? So in this way, linear regression could be useful for predicting protein stability. Okay. So again, in earlier class, we learned about the sensitivity, specificity, and such things, right? So here again, we are revisiting on that type of question, kind of, right? So it is asking that you have a particular method or an algorithm for which the sensitivity and specificity are given here. So the sensitivity is 95.5% and specificity is 85.5%. Now it is saying you that the label for your data, so the positive class and the negative class, those are switched. So your positive class has now become the negative class, and then the negative labeled class have now become the positive labeled ones, right? So then what would be the impact on the sensitivity and specificity? Will there even be an impact in this matter, or what kind of values that it would change to? So what do you think? If we remember the formula for sensitivity and specificity, it was something like this. So the sensitivity or the true positive rate was true positive divided by true positive plus false negative. So if you remember, that true positive is of course what the data was actually positive, and the system correctly predicted this data was actually as being actually positive, right? So let's say in a classification kind of sense. And then false negative was the data is actually positive, but the system is incorrectly evaluating it as a negative data. So out of all of that, what is the total fraction of the true positive data that was that the system could evaluate, kind of, right? So that was sensitivity or true positive rate. Conversely, specificity or true negative rate was true negative divided by true negative plus false positive. So out of all of the uh, actual negative data, what was the fraction of true negative? So that would kind of give you the percentage of specificity, right? So if we switch the labels for the positive label as well as the negative label, if we switch them around, so now the positive label become the negative label, the negative label again becomes the positive label. So what would the calculated values of these two parameters change to?
Excuse me, here we are switching the actual labels, but the predictive labels will be the same.
So you are switching the actual labels. That means that whatever you earlier classified as a positive data, now it will be classified as a negative data. And then whatever you had earlier classified as a positive data will be negative data, and then whatever you earlier classified as a negative data, now it will be a positive data. You have to consider it as a positive data. Okay. But the prediction was done already. So you are not re-predicting it, kind of, but you are just changing the label. So basically the positive label will become the negative label, and the negative label will become the positive label. So just that kind of switch. So try to answer it in the chat and just look at it. Okay. So here, if you notice, there is something interesting about the sensitivity and specificity formula. So basically, if your positive label becomes now the negative label, so that means the true positive, what kind of label we put this particular set of data at, what was true positive predicted earlier. Now, if we just change the label, so the true positive will become true negative, right? Because we are just changing positive to negative label. Conversely, true positive will become true negative, and conversely, the false negative will become false positive, because now the label is changed from negative to positive. So essentially, whatever you calculated sensitivity value earlier, now it will be true negative, kind of. If you go by the new label, it will be true negative divided by true negative plus false positive. Now, if you compare that with specificity, you will see that the original formula for specificity was just that, right? True negative divided by true negative plus false positive. So what is basically happening here is that if you change the positive label to negative label and the negative label to positive label, the value calculated for your sensitivity and specificity will also be switched. So in this case, since earlier your sensitivity was 95.5 and your specificity was 85.5%, now your sensitivity will become 85.5% and your specificity will become 95.5%, just from switching the labels. Because in this case, true positive will have become true negative, and then false negative will have become false positive. So it will basically mean true negative divided by true negative plus false positive. So that will be the value of specificity that you calculated earlier. Okay. So that means the sensitivity value will now be equal to the specificity value that we calculated earlier, and vice versa. the specificity value will now be the sensitivity value that we calculated earlier. So that was kind of the answer that the question was trying to get at. Just another exercise to practice this sort of formula for sensitiveness, specificity, and such. Okay. Now let's say on the same vein, if you are trying to now predict the class of a protein. Okay, so you have some sort of experimental protein, a new protein, and then you are trying to determine whether this protein belongs to this one class or this other class. So here exactly that is being asked. So if you have a protein X, a test protein, and you have two known kind of groups of proteins, proteins A and protein B, and you are asked that, okay, based on the amino acid composition between of X, A, and B, which of these following statement could be holding true? So if the correlation between X and A is higher than B, then should the protein belong to group A? So basically what all of the options are kind of trying to get at is that they are first basically telling us about an observation, and then after that it is making a statement, right? So you have to look at the observation and you have to tell whether that statement correctly represents that observation or not. Okay, so similarly in option B we had if X and A had a deviation value that was higher than X and B, so the protein should belong to A group, right? And then in C it is telling that, okay, if the deviation between X and A is higher than B, so X and A is higher deviation than X and B, and also the correlation between X and B is lower than X and A, then this protein should belong to group B. So which could be the answer maybe? So you have to first remember that what does correlation indicate? So if you have, let's say based on composition or something else, if you have a higher correlation, will that belong to that group or not? Or can you kind of expect it to belong to that group or not? And if you have a higher deviation, then can you expect that to belong to this particular group or not? Okay, so basically what does correlation indicate and what does deviation indicate? So in this particular case, if you look carefully, the option should be first one. So if you have two, let's say, components which are highly correlated, then you will say that there is some sort of connection between those, right? So in the terms of composition or in the context of composition that the question is asking, if you have X, the unknown protein, which has a higher correlation with A than B, then that will indicate that X is closely related to A and X is kind of not that closely related to B. So in this case, it will indicate if the correlation between X and A is higher, then the protein X will belong to group A. If you are just trying to infer that from this sort of setup, right? Let's say if you are looking at the other options, so in option B, it is saying the deviation term. So if deviation between X and A is higher than if you compare it with X and B, deviation basically indicates how distant it is from this particular group, right? So if X and A is having higher deviation, that will mean X and A is more distant than X and B. So in that sense, actually the statement were to be correct, it should be the opposite. So if X and A had a deviation higher than X and B, then the protein should belong to group B, which has a lower deviation with X. So of course, that is not what this option is saying, so it is not correct. For option C, again, it is saying that deviation between X and A is higher than B. So that means, of course, since X and A is having higher deviation, then it should belong to B. But there is another term or another kind of statement that need to be considered here. So it is also saying that correlation between X and B is lower than A. Now, of course, the first statement would have been correct, but if correlation between X and B was lower, then we cannot say that it belongs to group B, because the higher correlation was essentially group A, right? So in that sense, it should belong to group A. And if we have kind of conflicting statements from the first part and the second part here. So you cannot consider the option C, and of course you cannot consider all of the above in this case. So in this case, the option would have been option A, right? So in a lot of research problems, again, if you're trying to classify things into different sort of classes, groups, and such, so you might need to calculate sort of different parameters, let's say correlation, let's say deviation, something like that, and you have to particularly assign these sort of values or the the parameters that you are trying to calculate, or the, let's say in this case, the protein that you are trying to calculate the parameters for. So you have to assign those into different sort of classes or groups. So it is very useful to calculate these different types of correlation or deviation or these different statistical parameters basically. Okay. Uh, Now, again, so in algorithms, of course, they work on by solving a lot of functions and such. So let's say here we have very simple question. So you have been asked that a function fx equals SGN of something, SGN of x here. So here it is saying that, okay, so it is basically saying that you have been given these sort of different constants, let's say, right? So you have W0, you have W1, you have W2. So it is saying fx will be SGN of W0 plus W1 times x1 plus W2 times x2. So it is related to x1 and x2 by these different sort of constant values, kind of, right? So it is giving you the values for the W1, W0, W1, W2, x1, and x2. Then what will be the value of SGN of x? So here basically what you have to do, you have to calculate the fx, which will be SGN of this entire term from the given set of values, and then you have been given some sort of condition. So it will be 1 if x greater than equals 0, and it will be 0 if x is less than 0. So from that you have to basically calculate the value of SGN of x function from this condition. So first what you have to do is you have to calculate this term from the given set of values, and then after that you will have to just apply this condition here. Okay.
What is SGN?
It is just a random function, let's say. Okay, so it's just a random function where with its component kind of parameters you have to kind of calculate a value. Just a random function, let's say. For example, if you have heard of sin x, cos x, something like that, it's just a random function like that. Okay. And its value is kind of being determined by whatever inside it that we have. So you have to just apply this condition for SGNX in this calculated kind of value. Okay. So you can calculate. I think I see option answer B maybe in the chat. So in this case, what you have to do is that you have to calculate these terms. So W0 plus W1 times x1 plus W2 times x2. What it will become ultimately, if you calculate, it will become 2. Okay. So now you have the inside value corresponding to 2, right? Now, if the basically the condition was saying that if x was greater than equals to 0, which seems to be this case, then the value of SGNX, the value corresponding to this function, will be 1. Otherwise, it would have been 0. So since in this case we are talking about the inside value or x kind of as 2, then SGN of 2 will be 1. Okay. So in this case, f of x will be SGN of 2 equals to 1. So in that way, you can just simple numeric problem, you can calculate the value by applying the condition. Okay. Now again, it is saying about identification of co-expressing genes from mRNA expression data. That could be example of what? So I'll just briefly go over kind of this set of problems. So it was also present in one of the earlier iteration of the courses. So what they were talking about is the clustering algorithm. Okay. So binary classification is basically classifying into positives and negatives, as we talked about earlier, or 0 versus 1. So that's an algorithm for just classifying data into two categories, right? Non-linear regression, again, it is fitting certain dependent variables and trying to predict an independent variable, but it is not a linear sort of function. Clustering is again grouping of data between, let's say, into different set of clusters, right, or clumps, you can say. So if you have something like co-expressing gene data in terms of their expression level, something like that, then you can probably, let's say, group these mRNA expression data into different clusters, and then you can see whether, let's say, multiple genes are kind of having overlapping sets of clusters. You can do different kind of analysis regarding that, but that could be one way where you can find out about the information regarding co-expressing genes. Okay, so that is kind of what the question was trying to ask about. And then we had again another question which is called R square. So R, we know that was, the correlation coefficient, right? Pearson's correlation coefficient, that was R. But when it is R squared, so it is the basically notation for the coefficient of determination. And again, it is relating to the variance in the independent variable, how that is kind of expressed, and how you can get that from the dependent variable, or how you can get the independent variable data from the dependent variables expression side. So it is related to that, the coefficient of determination. Now with that, we have the sets of questions that we wanted to go through in this particular class. And now, as I said earlier, that we'll start with recapping whatever we went over from week 1 right up till week 12. So in this particular class, we'll try to cover some of that, and then in the next class, which is for tomorrow's class, we'll go through the rest of that. Okay. Yeah, I think I see one hand in the chat. Yeah, please go ahead.
Sir, I have one doubt.
Yeah.
Like previous class questions, I have to clear it. Can I go on?
Yeah, please.
Okay.
Go ahead.
Okay. Sir, spoke about Needleman alignment and Smith-Waterman alignment.
Yes.
So basically, the difference I want to clear. The difference is for Smith-Waterman, zero initialization is done, and for Needleman alignment, gap accumulation is done. Is that the main difference?
So for
-2, -4,
yes,
-6, -8. That is for Needleman.
yes.
And Waterman, zero. Everywhere, horizontally and vertically, it is zero.
It is 0. So basically, what is happening there for Needleman Wunsch, of course, you are trying to get the global alignment, and in that case, you need to accumulate the gaps further, right? -2, -4, -6.
Yes,
for
sir.
Smith-Waterman, the restriction is that if you are getting a -4 from one of the paths that you are tracing, then you cannot use like a minus value. You cannot put a negative value inside that box. So you have to put a 0 there. So from that, what is happening basically is that whenever you get a positive data, so in your entire sort of set of matrix, whenever you get a positive data, you can kind of get an idea, okay, my alignment for the local alignment algorithm is setting, it kind of settled there in a way, right? So I have, I think, these slides for Needleman-Wunsch in this recap slide as well.
Sir, I wanted to know,
yeah,
calculation purpose, for both we are doing the same, like vertical, horizontal, and diagonal. The system is the same except for one initialization is with zero, Smith-Waterman, and the other gap accumulation, Needleman with gap accumulation. Otherwise, the calculation is in the same way, no, sir?
Yeah, the process is the same. The process of getting to the optimal alignment is kind of the same. It's just that you are implementing it in a different way. Whereas in the
Okay.
Needleman-Wunsch algorithm, you had to put the gap scores for the horizontal and vertical jumps. Here you cannot do that. Here you have to put a 0. Uh, Basically, if you if you get a negative calculated score for a particular cell, you cannot put a negative value there. You can put a 0 there. That's it.
So, in both the cases, score is taken vertical, horizontal, and diagonal.
Yeah, yeah, yeah,
After
yeah.
calculating
Yes,
the
yes, yes.
the best score, the maximum score is taken.
Yes,
Right,
yes,
sir?
yes, 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 yes. So in all of these
Okay, sir.
cases, you have to trace through these different paths to find out, okay,
Hmm?
where you are getting your, where basically following which path you are getting to the next cell. So for all of that, you have to do. But if you're getting a kind of a negative score, you cannot put a negative score inside a cell. You have to put a positive, you have to put a 0 there basically instead.
Okay.
Hmm.
Thank you so much, sir.
Yes, I think I will recap that in probably this slide. I don't know if I have it. But I'll recap that in a couple of slides maybe. Okay.
Okay, sir.
Yeah.
Thank you
So
so much, sir.
yeah, yeah. So we'll just start from the very beginning. As we know that uh, for the very first class, Professor Brumha introduced briefly about bioinformatics and what kind of scope it covers. So we know that it is a sort of a converging point of these different fields of study, you can say. For example, we have math/statistics, which can give us basically the statistical insight or the mathematical insight into the data. If you want to know of the significance of your calculations and such, you have to rely on these statistical methods, right? Then you have computing, of course, the computing resources, etc. Those are needed for all of the calculations we do. You do huge amount of computing. Of course, we have IT, information technology, and last but not least, we have to rely on the fundamental sciences, right? So the main crux here, you can say, is the biology, because without biological data, there kind of would be no bioinformatics. However, biology is not alone in this fundamental science part. Uh, Bioinformatics or computational biology or anything you can think about related fields. So it also kind of uh, has relation to the other sciences, for example, physics, chemistry, the fundamental sciences, right? So the converging point of all of this, you can say, can be classified as bioinformatics. It is kind of an interdisciplinary field where it bridges all of these different parts and integrates the core sciences with the computer science and information technology and such things, right? Uh, what Where the biological systems complexity, so you can go right from the nucleotide sequence level to nucleotide structure level to expression level to now protein structure, protein sequence level, to even we can use the complex basically tissue level or cellular level processes, or even physiology or organism level processes. And in all of these kind of different levels of biological complexity, we can somehow implement the concepts of bioinformatics or the analysis of such to infer something meaningful. Basically, you can build a model to predict something or get insights into something from all of these different levels. So that is what kind of this slide was trying to say. And of course, if we go by the prominent functions of bioinformatics as it is seen, so from structural analysis to sequence analysis to function analysis, in all of these different cases, the bioinformatics application can be classified into. Of course, for example, in structural analysis, we know protein structure prediction is a huge thing. We know that 2024 chemistry Nobel Prize, basically half of that was given to jointly to the AlphaFold teams, John Jumper and Demis Hassabis, we know, for their AlphaFold remarkable achievement in the protein structure prediction field. So that was one thing. We also know that David Baker was awarded the prize for functional protein design. That is again one other avenue that we can even explore in this entire bioinformatics computational field. Not only structural aspect analysis, but of course you have the sequence. You can analyze the sequence to get meaningful information. For example, we talked about sequence-based parameters. We can use the sequences to get phylogenetic information about evolutionary distances and such things, right? So different kind of analysis can be done. Of course, functional analysis is also one path that can be explored hugely here, right? And ultimately, combining all of that, we have very important software development and database construction and curation, because those will be what sort of collects everything from the analysis regarding the data that was gathered through the analysis. We can put the analysis algorithms in a web server and allow that to access, allow different research groups or anyone who is interested in that to access the particular server and the tools that we have built up. And conversely, you can develop different softwares and you can distribute them through different sources, and we can allow different groups to perform their own analysis and own uh, sort of from their own research data through those software. So software development and of course database construction are a hugely important part of this entire bioinformatics sort of field, right? And a lot of important advancements have uh, their roots in sort of these territory, basically the databases and the software development as well. Of course, without these, we would not have the UniProt, we would not have the protein data bank and such things. We would not have AlphaFold 2 if it was not, it could not be that much useful if it was not distributed widely, right? So those are very important. Uh, if we talk briefly about the significance of studying bioinformatics, again, the primary objective is to enhance comprehension at the cellular level functions or the molecular level functions by examining sequence and structural data, then utilizing the structural and sequential approaches to solve functional problems, then fundamental genomic and molecular biology research, knowledge-driven drug design, forensic DNA analysis, etc., computer-aided drug discovery, analyzing protein ligand interactions through the use of different potentials, right? In classical mechanics, the different potentials can be used in that way. So it can facilitate basically the discovery of drugs by decades, because we are kind of screening the available huge libraries, and then we are trying to narrow down the path of discovering a compound that targets a specific protein, let's say, right? So that can save a lot of resources and a lot of time as well. Then we have the knowledge of protein structures, which can, of course, define high affinity specific targets, which we talked about earlier. Also in photosynthesis, we have molecular phylogenetic analysis, genomics and bioinformatics. For personal healthcare, that is also one important avenue, etc. Agricultural benefits are also quite widely used, and it is of high significance, basically, right? So the main 5 parts that Professor Gromiha kind of talked about, what the entire scope of bioinformatics in terms of their applications cover. So firstly, you have the databases, construction of databases. Then you have the computational derivation and testing of hypotheses. Then you have web servers, where you have built some tools and put it on a server for others to access. You have virtual screening and drug discovery, which is the drug discovery / development pathway. So it is the very first part of a drug discovery pipeline. And then we have conversely big data analysis, for example, in next gen sequencing. We get a lot of data that are kind of produced, and to analyze all of this data, we will have to look for some efficient processes, which bioinformatics can give us that opportunity, basically, right? So we talked about the different sort of databases that are very important in this field. For example, we talked about the structural information data, which can be obtained from this RCSP PDB database. We talked about the other ones as well. For example, GenBank, DDBJ, AMBL, they have the sequence related data for genes or DNAs, right? So DNA sequence related data can be obtained from those. And from the sequence related data, so we can basically use that to know more about the biomolecules that you're trying to investigate. So one of that could be sequence related or sequence based features. Another one could be, for example, very simple one like composition or possibly charged amino acid, composition of non-polar amino acid, composition of let's say the structural components or structural elements, right? So what part of it is kind of dominant by beta sheets, alpha helices, and so on. So this very similar, very simple compositional analysis can be done from just the amino acid sequence. I guess for the structural ones, you need the structural data as well. But for sequence-based analysis, you can do these charged amino acids versus non-charged amino acids, polar versus non-polar amino acids, and so on. These kind of compositional studies you can do easily. We look at some example from this perspective. And then the big data analysis that we talked about earlier, of course, in the earlier era, basically the Sanger method was widely used. Now you have the NGS sequencing processes which can churn out a lot of data in a small amount of time. We saw one plot is that from, let's say, 2001 right up to 2022. I have not the updated. I do not have the updated data here, but you will see that the cost of raw megabase per DNA sequence at certain point, for example, around this 2007 kind of mark, it started falling very rapidly, right? So this sort of point indicates uh, the next generation sequencing processes that was kind of being developed at that time, which uh, accelerated the progression in this field by a landslide, kind of, you can say, right? For example, if you compare the Moore's law, which is kind of an indicator of progression in a field, you can say it is related to the number of kind of density of transistors in an integrated circuit. So in the Moore's law curve, you can compare uh, the curve for that, and you can kind of say that okay, so it kind of doubles in 2 years. So if you have sort of progress in some field which is staying inside this Moore's law curve, you can say that the progress is kind of okay. It is steadily progressing, but however, you have this 2007 mark where a huge kind of improvement was upon us because of the NGS sort of approaches that were developed at that time. And in this plot, whenever you see a valley or a sharp decrease in the cost of raw megabase per cost per raw megabase, you can expect that okay, at some point, at that point, probably some important improvements over the existing approaches for NGS was kind of performed, right, or discovered, basically. So same thing, if you can represent in different terms, you can see cost per human genome sequencing, that also decreased rapidly. So from that, you can say that, okay, so since the cost is decreasing that much, the time for uh, performing the analysis is decreasing that much. That means it must generate a lot of data, right? A lot of data, which of course can be efficiently analyzed through bioinformatics. That is the point that this slide was kind of telling us, right? So from NGS, you can do, of course, the data analysis. You can do genomic assembly analysis, functional annotation. You can compare genomic data, transcriptomics, epigenomics, pharmacogenomics. So relating that with pharmacological kind of data. So how genetic variation can influence the performance of certain drugs, those kind of data, those kind of analysis, and also personalized medicine. So all of that can be kind of related to NGS, and of course, for analyzing the data, you need bioinformatics, right? So for sequence-based sort of analysis that we can do, that we talked about earlier, also like compositional studies and so on, we can actually effectively make different sequence-based features. So any feature for proteins which does have some decent link with its sequence, we can basically express that in terms of the sequence-based features. So where we basically take small components of the portion of the sequence, for example, a subsequence, it could be a 2 nucleotide sequence or 3 nucleotide sequence or something like that, and you can relate the available feature through that sort of set of sequences. And how we can basically get that? So for example, if we have this long stretch of nucleotide sequence, so we can get the number of overlapping trinucleotides and the overlapping dinucleotides from that. So if you have n equals 13 as the total number of components present in this sequence, you will get n minus 2 equals to 11 number of trinucleotides from this, and you will also get n minus 1 equals 12 number of dinucleotides from this. So basically, the overlapping trinucleotides and dinucleotides. For example, if you start from this particular A, you can go forward 2 steps. So AGC can be one trinucleotide that you can use to correlate. You can use to calculate some sort of data or feature in terms of this particular trinucleotides, right? So AGC, GCA, then CAT, then ATG. So these will be your entire stretches of trinucleotides, overlapping trinucleotides that you can get. And if you have any data or parameter related to these sort of trinucleotides, then you can say, okay, you can calculate the entire stretch of sequences parameter from those kind of individual components. So that is kind of the point here. A very good example here could be, let's say we have the hydrogen bond energy for the sequence AT-AGAT, and we have been given that, okay, the energy for AT is -7, TA is -7, AG is -9, and so on. So what you can do is you can decompose this entire sequence into overlapping dinucleotides and you can just add up all of these overlapping dinucleotides value. You can get an average, and that will give you an ultimate value of minus 7.80. So that could be a handy way of proceeding with this sort of calculations, right? Same thing here. You have been given the different hydrogen bonding energy for the individual components. You can divide this sequence into overlapping dinucleotides and ultimately get an average to indicate the actual value for your sequence, right? We learned about some simple concepts, for example, GC content and so on. You can just get the total number of GC. Conversely, you can get the total number of AT, and you can just divide by the total number of nucleotide components that you have in the sequence and multiply by 100 to get the GC content and such calculations, right? So now we are talking about basically sequence-based parameters in a bit more detail. So earlier you talked about from the overlapping dinucleotides and trinucleotides, you can calculate the individual values for certain parameters for those dinucleotides or trinucleotides, and then you can get the value for your entire sequences, right? For example, these values that you can kind of calculate could be DNA rigidity, it could be thermal stability, it could be base stacking energy, it could be hydrogen bond energy, and so on. And the number of nucleotide steps that you decide to use here, so that needs a particular balance. So if you have a mononucleotide sequence, so if you are just trying to calculate based on, let's say, just single nucleotide data, versus if you are just trying to calculate based on a trinucleotide step, overlapping trinucleotide step, you will see that some parameters are kind of changing between them. So the more that you go from mononucleotides to the higher kind of order of subsequence level, so let's say dinucleotide sequence, trinucleotide sequence, and so on, you will know that the information increases. What you can get from the data, that information increases. Now, why is that? So let's take a quick example. Let's say if you have a 100 length sequence, right? Nucleotide sequence. It could be something like AGT, CGA, GA, something like that. Right? Let's say 100 components are there. If you are only calculating data for mononucleotides, so of course you can have, let's say, calculation for adenine, you can have calculation for guanine, you can have calculation for thymine and for cytosine as well. And let's say you just have these 4 values, and you can put these 4 values in different compositions or for whatever your sequence dictates, and you can get the total number of value from this particular data, right? But imagine if you go just a little bit higher step. Now, whereas in mononucleotide calculations, your total number of individual components that you're adding up was only 4. In case of dinucleotide sequences, you will see that you can generate a lot more individual components, right? For example, in case of dinucleotide, you can have AT, you can have TA, you can have AG, you can have CG, you can have TG, you can have GT, something like that, right? So a large number of sequences, more number of individual components than the mononucleotide was obtained here. So the quality of information that you're trying to associate, that you're trying to kind of predict or calculate, will increase because the associated number of individual components was increased. Okay, so the quality and the nuance of information that you are getting from that will increase. However, if you go too far, for example, in trinucleotide sequence, and if you go even further, let's say tetranucleotide sequences, what will happen is that since you can generate a large number of tetranucleotide sequences in total, right? So it will be a large number of tetranucleotide sequences, and of course, if you have statistically significant data for each of these tetranucleotide sequences, then of course you are very good. But while you are trying to determine the individual values for these tetranucleotide or trinucleotide sequences, you will notice that the number of samples or data points that you are getting for this particular sequences, some of them will be very low. So for those set of sequences, your statistical significance will decrease by very much. So because the available data decreases, the number of sequences basically you increase. So that can reduce the statistical significance of some of the individual components, which can affect the data. So there needs to be a balance between them. For example, here Professor Gromi has shared this slide. So we have, of course, on the left-hand side, the dinucleotide sequence and their related parameters like twist, stacking energy, rise, bend, dip, inclination, major group width, etc., right? On the right-hand side, you will see that trinucleotide sequences in this paper when it was published. So they also calculated the trinucleotide sequences and their associated parameters. like Young's modulus, like that, right? This is a part of like Young's modulus. Now the number of samples that they used, so they of course used some database to determine the values for the individual trinucleotides, but you will notice that in some cases, for example, in this ACA TGT, so this ACA trinucleotide, for that they only got one sample where they could basically get, they could calculate the data from. So from that you can see, okay, in this trinucleotide sequence only you are getting only one sampled data from which you are trying to obtain the value for the ACA trinucleotide. So that means the statistical significance of this particular component will be much lower, because you are only getting this data from only one particular sort of sample, right? Conversely, if you saw that AAT that had a large number of samples, so 49 samples, so of course you will get statistically significant data from that. But for something like this ACA, something like CCG, something like CCC, you will not get that much data to confidently say that this is the actual value for this trinucleotide. So the further you increase your nucleotide steps for calculations, the more you will be in this sort of situations where you will not get significant data for the individual components, basically. So there needs to be a balance. So generally, in this prediction processes, calculation processes, dinucleotide and trinucleotide sequences are considered good, basically, right? So again, we looked at the different numeric questions that can be obtained from that. Of course, in the earlier one also, we thought that if we had n equals 13, then the number of trinucleotide steps can be obtained from the formula n minus 2. Conversely, the number of dinucleotide steps or sequences can be obtained from n minus 1, right? And you can calculate different parameters based on that through these very simple numeric sort of problems, right? Uh, so that was sequence-based parameters, which is again one important kind of part of the whole field. Now, another part, as we talked about, was databases, right? So database is basically a repository where we design and store data, uh, designated to store data. But not only that, it has to be organized in a manner that facilitates easy retrieval, and it has to have some sort of tools which allows custom retrieval of certain data and more efficient retrieval of certain data. So, for example, you cannot just feed the entire data set that you have to the person who is kind of trying to give the query, right? So, from their particular selection, their particular criteria that they're mentioning in the query, from that you have to give them, or you have to provide the output for very specific set of data. So that is why it is also important for them to have some advanced search criteria, right? So it is basically the databases kind of comprising both the hardware and software components for effective data management. And the primary goal is to uh, organize and structure data into easily retrievable records, kind of, right? So, for example, here we have a simple example, like it can contain multiple fields. So these fields will indicate, we have a column that kind of dictates a particular data which a user might be interested in, right? We have the different sort of characteristics or terms regarding databases. For example, you have content, we have the ontology, which is the list of valid terms. We have the schema or the logical structure or the representation of the data set, the data format, the retrieval methods, and then links also. So linking to other external resources or databases or publications and such things. So they are also important for the databases, right? For biological databases, we know that they can be classified into three main types. One is primary databases, another is secondary databases, another are specialized databases. So primary databases are basically storing original data, for example, raw sequences or raw structural data that are submitted by the community. If you look at the example, you will get the GenBank, European Molecular Biology Laboratory, EMPL, DDBJ, DNA Data Bank of Japan, and as well as Protein Data Bank, which stores the structural data. So these can be classified under the primary databases. And the secondary databases are derivatives. Okay. So they are kind of the processed or manually curated information that are derived from the primary databases. So for example, SwissProt or Protein Information Resource, those can be classified as secondary database. And then if you have a specialized database which only have data on certain niche topics or a very narrow set of coverage these databases have. For example, you could have a HIV sequence database, so it stores the data related to only that. So these are the specialized database. We looked at SQL or SQL, structured query language, which is one sort of programming language you can say, which is very helpful in creating this database and then retrieving information from this database as well. So for construction and retrieval and everything, structured query language is very useful. So it is specialized programming language for that thing only, for managing and manipulating relational database. What are relational database? We will just look into it. So the different categories of SQL command are there. For example, select, insert, etc. They can be classified under different categories: data query language, data control language, and such. So a relational database, what we talked about earlier, where the SQL is used basically. So they kind of are organized in a way that different tables are given, and these different tables have at least one common attribute or at least one column, common column between them, so that you can easily correlate the data for one table to another table. Okay, so these have certain benefits over a flat file database, which we'll just see in the next slide. But the relational database, as it says, so it is a part of a database management program or database management system. So these systems are basically developed to streamline the organization and search and retrieval of data, and they also incorporate the operational instruction for the database basically. Now, what type of database management systems are there? Of course, as we talked about earlier, relational database management system is there, object-oriented database management system is there, and the databases that implement those are classified as relational database and object-oriented database and such, right? So relational database is, of course, structuring the data into multiple tables with at least one common attribute for efficient data organization, right? So here each table is also termed as a relation. They are composed of the rows and columns. Columns are basically the individual field where you will have multiple data records, and then the rows are the corresponding values that are recorded for that field. You have good indexing, then you have query execution as well. For example, the system selects linked data from different tables during the execution of the query. You have certain advantages over a flat file database. So flat file database, just next slide, I think one example is there, is where you just have the data in a flat file where no hidden instruction for organizing or for retrieval is given in this kind of file. Okay, so we'll notice that in flat file database, a algorithm has to go through the entire data file to retrieve whatever we want it to give the output as, but that will take a long time if it is like let's say thousands of lines of data or thousands of rows of data that it has in a flat file format. So it will take a long time to retrieve that data. For relational database and object-oriented database and such, the efficient organization and the instruction that are kind of operating underlying of the database, that can help us quick and easy retrieval as compared to the flat file format. Of course, it uses the relational databases use SQL or SQL for the particular organization and retrieval and such things. Right? So here, for example, we had the flat file format example. So it just states that name states course number and course name excuse me, for certain courses, right? So we just have pipe or a delimiter kind of classifying the different records that will go into these different sort of attributes or fields. But if you are trying to ask a complex query, for example, give us the courses that the students from this particular state, let's say Texas, are taking, right? So for that, the algorithm will have to go through the entire flat file because there is no hidden instruction, and then ultimately, let's say it retrieves something, right? If you compare that with something like the relational database where it is given in a table format, so all it has to do that it has to read the table one entirely, and you can see that the state information is given there. It has to retrieve the state information corresponding to our query, the student ID here, the student ID corresponding to our state information query. And then after that, for table 2 and table B and table C, it does not need to go through the entire tables. It can only relate the student ID with the course ID and then the course ID with the course name, which will give us the output in a much less time, basically. So that is the advantage of relational database in terms of retrieval of data. We have again object-oriented databases where the relational databases kind of cannot represent the hierarchical relationship in a good way. So that is why the object-oriented databases were kind of created. So they use the object-oriented language such as C++ and link object using something called pointers, right? So these pointers will be kind of be pointing one object to another. We'll see in the next sort of slide. And of course, since relational databases also have some rigorous mathematical foundation underlying them, so combining the good part of relational or the advantages of relational databases with object-oriented databases, giving us hybrid approach for organizing and creating databases, which are most modern systems kind of employ, right? So here we have the object-oriented database information where the state object kind of have different components. You can say state equals Texas, and then it is kind of linking this particular object to this sort of course object or student object, and then after that the student object is linking to this course object. So these sort of different pointers can help organizing and retrieval of data also in an efficient manner. Now we went over big data analysis, we went over the database construction curation, and the very important concept was drug design, right? So we know how we design drug is that we are trying to interfere or modulate some sort of cellular process or cellular signaling. And we are hoping that by modulating or interfering with that signaling, whatever disease symptom was showing earlier, it will be alleviated. So that is the kind of the principal reason for the reason behind these drug discovery approaches that we do. So for an example we can give is that if some inhibitor is designed for a protein pocket where that pocket needed to be binding to let's say an external agent or a physiological agent that ultimately manifests in some disease conditions, so you can design an inhibitor against that pocket and it will bind to that pocket, and what it will do that it will not let the actual physiological binding partner or the diseased state binding partner to insert into that pocket. So that will mean that the signaling cannot proceed forward, and then we will have relieved symptoms basically, right? So that is a treatment for the different diseases. So in drug discovery, the role of computational biology is basically to identify potential lead molecules. So computational biology serves as the initial part of the entire pipeline for drug discovery, where you are narrowing down from a large search space of different molecular structures, small molecular structures. You are narrowing down to a particular small set of compounds that you can ultimately proceed forward with your wet lab experiments and clinical trials and such, right? So it can identify potential lead molecules that can be proceeded further for the experimental studies. So that is the role here for computational study in drug discovery. Okay. Now we looked at the different concepts regarding sequence alignment. Now, why sequence alignment are needed? Of course, we know that basically sequence alignment can give us an idea about the sequence similarities, and let's say the sequence similarities can again further give us ideas about the core functional part of some proteins, right? So if you have some unknown sequence that you have gotten from your lab and you are aligning that with the already available highly annotated set of sequences that are available, right? So then if you get a match, if you get a good match that your new sequence is kind of very highly similar to this other one, then you can say, okay, so this other one had this function that was defined for this particular protein, and now, or protein or nucleic acid, and now you are getting this new nucleic acid or protein that is highly matching with that. So you can probably infer that, okay, so it could again do very similar set of functions to that previous annotated ones, right? So from that sequence similarity, you can get different information. Suppose it would be useful. You could generate phylogenetic trees. So these trees represent the evolutionary relationships between different species. You can get that information. You can do homology modeling for protein structures through the use of sequence alignment. Let's say you have some protein where the structure is not determined yet, or if you have certain small region of a protein where the structure could not be well defined in a crystal structure, then you can align that sequence for that protein with some other sequence where the structure is already available. And uh, based on the aligned portions, you can kind of get the structural information. You can copy a lot of structural information and infer a lot of structural information from the already available structure for the sequence that you aligned your new sequence to, right? So you can get that analysis, you can perform that analysis via homology modeling, and for that also sequence alignment is required. Of course, inferring the function of newly sequenced genes is important. Uh, and then in global alignment, of course, the overall similarity and evolutionary distances and such things can be obtained basically, right? For local alignment, it can help you in discovery of small motifs or sub-motifs that are present within a protein, uh, which can again give us idea about the particular function or conservedness of that protein in a sense, right? For structural prediction also it is important. So uh, one very simple way of visualizing sequence alignment we went over was something called dot matrix. So it was kind of introduced in 1970 as a novel method for comparing amino acid and nucleotide sequences. So it is basically constructing a graph where you have on two sides uh, two sequences kind of written, and then you have to put a dot wherever you see a matching character in the form of a matrix, right? And from the dots that are kind of put inside this plot, you can try to infer the different patterns. Basically, here you are trying to look for the patterns of diagonals, which can give you the information about similarities. Okay. So very quick example is kind of given here. So on the top hand side, we have seen the let's say sequence 1, right? It has something like 1 to 220, 240, something like that number of nucleotide side. And then we have another sequence which is on the left hand side, right? And to compare these two sequences to see where they're matching, what you do is that you just put a dot in between all of the matching characters, and ultimately you try to see observation where are you finding the diagonals, right? So the diagonals can give us the information about the matching region, which we will see just a moment. So here we had one example. For example, on the top hand side, we had ACGCG. On the left hand side, we had ACACG. Now what we have to do to fill up the dot matrix, we'll have to just put a dot wherever there is a visible match between the characters. For example, A versus A is matching here, C versus C is matching here, again C versus C will be matching here, and so on. So if you put all of this matching in the entire dot matrix, you will see that that a diagonal was kind of formed in this particular portion, right? If you see this diagonal, and if you project this diagonal, that the one that I'm currently kind of hovering over with my cursor, if you project this diagonal onto the left hand and the upper hand axis, you will see that they both correspond to a ACG subsequence in sequence 1 and sequence 2 also, right? So that means that from this dot matrix, you can see this ACG subsequence is matching or aligning together because both of them has ACG, as you can notice. Now, this method of dot matrix, we had used a window size of 1 and stringency of 1. So what window size particularly indicates is that how many number of components you are willing to compare at one particular time. So if we had, for example, a window size of 3, instead of matching one character by one character, what we have done is that we would have matched the first 3 characters of the first sequence with the first 3 character of the second sequence. And if for the stringency of 2, if we had 2 exact positional matches, for example, in this case, if you compare the first 3 sort of subsequences, if you see that their very first 2 components A, C are matching exactly, right? So we have at least 2 exact positional matches. Then you can put a dot between these 3 sort of subsequences in this position. Okay. Now after that, you have to proceed forward. You have to slide this window forward just a little bit. It will be overlapping windows. You can make that overlapping windows. You can set the window step size also, where in the next step maybe you are not exactly sliding the window one step forward, but you can manipulate it to slide it, let's say 2 or 3 steps forward even. So you can choose that by yourself and see what kind of gives you the best results. But in this case, if you match the 3 by 3 sort of subsequence or windows here, ultimately you could have gotten something like uh, this kind of plot, right? Now, in this particular case, you will see that this, if you just visually look at this one, so on both hand side, you see the projection is kind of not matching. It is CGC versus CGC, CGC versus CAC, not exact match, quite similar, but it was very kind of, you could argue that it is probably not uh, that better match than what we had in the window size one and stringency one, but that is because here the number of components in the sequences was very low, right? So if you look at a very large, let's say sequence versus sequence sort of match or dots, you will see that if you just start matching character by character, you will see a bunch of dots in your plot, where a lot of times inferring certain patterns from this dot plot would be very hard or very challenging, right? So for that only the window size and stringency and the window step sizes were invented or kind of used. One good example would be here. So from the left hand plot, you will see a character by character match is putting a lot of dots inside this plot. Probably you cannot find a good pattern that you can see in this plot. On the right hand side, of course, you will see that the number of dots that this plot is showing is much less. However, there seems to be some sort of pattern present here. So this was done with a window size of 23 and a stringency of 7. So out of 23 step component, taking that as a window, if 7 of them had kind of the exact positional match, then that was counted as a dot in this particular plot on the right hand side plot. And in this particular case, you are seeing some sort of patterns. And what interesting is about this particular plot is that it is a same sequence versus same sequence plot. Now, if you compare same sequence, let's say sequence 1 versus sequence 2, different sequences together, then of course you will see which portion of these different sequences are matching together, where they are aligning together. You can get that information. But when you match a same sequence versus same sequence data, what information can give us, it can give us is that it can predict repeated motifs basically. So you will see on the right hand side plot B is that on this very initial part of the sequence, you can take either one of them. You will see that a lot of basically diagonals on the small diagonals on the opposite sequence that is kind of observed in this region, right? So these are indicative of repeated motifs. So one single stretch of amino acids or similar amino acids that was present repeatedly inside this sequence, right? So these repeated sort of subsequences can be observed or identified by comparing the patterns, basically comparing the same sequence versus same sequence and manipulating the different window sizes and stringency and so on could be very useful in certain studies, basically, right? Now,
Excuse me,
yeah,
in the previous example.
yeah,
So here you told ki window size is 23 and stringency is 7.
yeah.
So the dot will be placed exactly in the center of that 23 window size?
Yes, yes, yes. The dot will be placed
Exactly.
exactly at the center of this 23 window size. Yeah.
Okay. And so after 23, so here obviously the step size will be greater than 1, right? Otherwise, this much clear plot
Yeah,
will
yeah, yeah.
not come.
Yes, yes, yes. So I am only saying 23/7 because it is written here kind of Windows 23 and stringency of 7, sorry. I don't think they have mentioned the step size here, but yeah, step size will be higher than 1 basically, yeah. Because otherwise it could have been a lot more kind of cluttered, you can say, right? A lot more dots should have been present, yeah. Yeah. Yeah, so that was kind of a visual representation of dot plots and how we can infer the patterns or alignment patterns from kind of these sort of settings, right? However, this is only visualization kind of approach. For properly aligning different sequences, you need a more rigorous setup. By what I mean is that you need to start scoring each of the alignments, for example, each of the alignment components like matches, mismatches, or wherever there is gap present via certain scoring procedures, right? So that can give us an idea about the overall kind of score of the alignment. And if you are comparing different alignments and so on, this can help us get a better idea at, okay, which alignment is the most probable kind of, right? So that is very useful. And here we, in the simple kind of examples we had, for example, for aligning these different sequences, match score of 2, mismatch score of 1, and negative gap score of -2. Sorry, match of 2, mismatch of -1, and gap of -2. Now, why is that? Match score, if your characters are matching between two sequences, of course, that is very positive, because that means the alignment is working well. So you are kind of rewarding that sort of alignment in the scoring process. If your characters are not matching or mismatching, uh, so that you have to kind of negatively penalize as a score, because ultimately you want to get the most optimal alignment as where your most of the characters are kind of matching, right? That's why the mismatches are negatively penalized, and then the gap is even more higher, negatively penalized, because in evolutionary processes gaps are kind of rarely generated. Okay, so if you are seeing in an alignment that there is a certain gap, so that means that the probability of that happening naturally through the evolutionary process is much lower. So for that, if you are seeing a gap, then you have to penalize it with a higher sort of negative score. That is why the gap score you will see mostly, they have a higher negative value than mismatch scores, let's say, right? For these sort of simple kind of setups, uh, for this sort of alignment, again you can just calculate the number of matches versus mismatches versus the gaps, and you can get the optimum alignment score as the net score here. For example, in this case it was -1, right? Let's say you are comparing two sequences, you are trying to align two sequences, and you are getting three different possible alignments. Here we have the left hand side, middle hand side, and the right hand side. And here again we are introducing another other penalty basically, which is called the origination penalty in this case. Now, earlier we saw the gap penalty. Here it is divided into two parts. The length penalty is for the length of the gaps, and then the newly introduced origination penalty is that wherever you have a new gap introduced at this sequence alignment, you are again trying to penalize that, because through the evolutionary process again, new gap introduction is kind of much rarer, right? So that's why they are highly penalized. Now, let's say you have these three different types of alignments that you think could be possible. So what you could do is that you could try calculating uh, the net alignment score from the different match versus mismatch versus the length penalty and origination penalty scores, and you can get the optimum, you can get the alignment scores for all of these three different alignments. So, for example, the left hand side was -3, the middle one was -1, and then the right hand side was +1. So in this case, the +1 would be the best alignment score, and as such, you can say that the alignment for the left-hand side would be the best, right? So that was one way that you can kind of identify optimal alignments. Of course, the scoring setups are not always this simple. So in actual cases, the scores that you need to determine should come from some well-established data, right? For example, such as different matrices that are used. For example, PAM matrix, Poisson matrices, and so on. So those can give you very rigorous kind of evaluation of particular match versus match and particular mismatches between amino acids. It can clearly sort of define those sort of scores from either observable data or either interpolation from some matrix data, right? So that was the importance of PAM matrix. Now, what PAM matrix basically was is that it was originally constructed by Margaret Dayhoff. And what she and her group did is that 71 groups of closely related protein sequences were taken, and they, used a cutoff of greater than 85% similarity, greater than equal to 85% similarity for this, for determining which could be classified as closely related proteins. And they sort of made some assumptions. For example, that they kind of thought that mutations in closely related sequences would not significantly change the common function and such. And whatever mutations are observed in nature, that can be classified as natural selection, or that can be classified as accepted mutations. So that's why the matrix got its name from point accepted mutation, or PAM matrix. Now, next they did a phylogenetic deconstruction, and after that the PAM matrix code derivation was done. So in the PAM matrix, basically one PAM unit is defined as 1% of amino acid positions that have undergone changes. So that could be classified as one PAM unit. So that means, let's see, if you have a PAM score of 120, that means 120% of amino acid positions have undergone changes. Of course, one single amino acid positions could have undergone through different set of intermediate changes and ultimately reached a, let's say, final stage, right? So it can happen like that also. And after that, when you have basically constructed the scores for the lower level PAM matrix such as PAM1, so PAM1 will indicate the closely related protein sequences score, because they are only using the closely related protein sequence to uh, get the data for the PAM1 matrix, right? Now you can get the higher order PAM matrices individual component data, so protein by amino acid change or mutation data. What you can get that from is that you can multiply the lower level PAM matrices. For example, if you multiply PAM matrix 1 with a PAM matrix of 1, you will get the data for the PAM2 matrix. Okay. And you will notice that if you keep multiplying these things, the probability of your, let's say, some amino acid, let's say alanine, staying the same will get reduced slow by slow in every subsequent multiplication. Conversely, let's say, for example, if you have just like a simple example, if you have like an alanine to in a closely related sequences, you saw that okay, alanine is staying alanine, let's say 98% of time, and the other 2% of times the alanine is kind of mutating to different amino acids, right, for a certain position. Okay, that's like for overall, let's say the overall proteins, right? Now, what you can do is that you can then construct the matrix where you put the alanine versus all of the other 19 amino acid, including the alanine itself. So you put alanine versus all of the 20 amino acids, and you just put the data of probability of it is staying same or changing to that, right? So alanine versus alanine will be kind of like 0.98, because the 0.2 will be distributed among the other amino acids versus alanine junction, right? Now, if you multiply that matrix with the same matrix, you will notice that in every subsequent multiplication step, alanine versus alanine score will slowly start to reduce, and the other ones, the mutation scores, that will slowly start to increase. So that is just a nature of the matrix multiplication in this case. So from that, you will notice is that if you just keep increasing the kind of the level of the PAM matrices, so if you let's say do PAM 40, so you are multiplying PAM 1 by itself 40 times. Let's say you are doing PAM 100, you are multiplying PAM 1 by itself 100 times. You will notice that in all of these cases, essentially what you are getting at these highly higher level PAM matrices is that you are getting data for structurally or sequentially divergent set of proteins. Okay, so data for sequentially divergent set of proteins, that prediction you are getting from these higher level or higher order PAM matrices, because in those cases you will notice that the alanine staying alanine, that probability was much reduced, and then the other ones, the mutation probability was much increased. So that is why when you are trying to compare, let's say, very closely related set of sequences, it is better to use a lower level PAM matrix. Let's say PAM 40 you can use, for example, something like that. If you are trying to use, if you are trying to analyze or align structurally divergent or sequentially divergent set of organisms, then for that you have to do, you have to take a higher order PAM matrix as your baseline, basically. So that is kind of the PAM matrix part. Uh, of course, ultimately the score is converted to log odds score, and the interpretation you can kind of get in something like this. So if it has a PAM1 number, PAM1 matrix, so that means the observed mutation rate is 1%. So 1% of the positions in the 1% components of the sequence is kind of changing, right? So sequence identity remains 99%. Similarly, if you go to a higher level PAM matrix, for example, PAM 250, you will see that the observed mutation rate is much higher, 80% versus the sequence identity, which is only 20%. So that means structurally divergent, because only structurally divergent or sequentially divergent uh, organisms will have, if you compare their the gene and so on, they will only have this sort of high mutation rate between them, right? Because it is changing that much. That is why it is becoming divergent, right? So for closely related species, you have to use a lower PAM number. For divergent kind of species, you have to use a higher PAM number for this match score and mismatch scores predictions and so on. So here is some simple, basically in uh, PAM matrix demonstration kind of. So you will see that the different scores are kind of indicating, in a way, the probability of this particular amino acid staying the same or different, or and such. For example, if you just look at, let's say, what should we look at? Let's say that if you just notice the corner, so for these corners, you will see that the same amino acid versus same amino acid data is kind of given. So the positive data that it is kind of indicating, that means that this amino acid staying same, that score is calculated uh, as the positive score, because of course in a lot of times it will not mutate, right? However, if you see certain amino acid to amino acid mutation that has a highly negative score, so that means that those mutations are not kind of accepted by evolutionary processes and they can be penalized because it is a negative score, right? So in your entire alignment scoring, you have to put this particular score from this particular mismatch, let's say. So that is the usefulness of PAM matrix. And Blossom matrix was another matrix, which is the blocks amino acid substitution matrix. Yeah, I see one hand. Yeah, please go ahead.
Sir, please, the previous slide once more.
Yeah.
Sir,
So,
here, sir, 1 second, sir. Here, what does this C, first thing, C12 and 17 particularly indicate, sir?
so the 12 to 17, yeah. So this 12 to 17 is basically, if you notice on their both axes of projections, you will see that they indicate the scoring for the same amino acid. For example, this 4 here that I'm highlighting, it is indicating a Q versus Q score. So that means that it is basically indicating the probability of a Q kind of staying the same, or a glutamine kind of staying the same. So that is it is trying to indicate. For example, if you take a look at the C versus C12, so that means that the log score was calculated as such that this score is representing the probability in a way that the cysteine is staying cysteine. It is staying the same. Whereas all the other ones will indicate that the cysteine mutating to this particular residue. Okay.
Sir, so you mean to say it stays for a longer time, more the integer, stays as such for a longer time.
Yes. So the more the integer, more the positive value, that means that this amino acid to amino acid substitution, or if it is staying the same, so that means that process is highly evolutionarily favorable. Okay. So even if you see, for example, let's say serine versus alanine, you get a positive score. That means this substitution is not that penalized in this scoring system. So this substitution is evolutionarily feasible. Okay. However, if you see a highly negative score, for example, C versus G -3, or let's say C versus D here -5, so that means that is not favorable by evolution. As such, we need to penalize it. And where did we get this data from? We get this data from the PAM matrix construction, where this is a PAM 250 matrix. So basically, those closely related sequences were compared in PAM1 matrix, and then from matrix multiplication, we are kind of interpreting the data for it could be for PAM 250 matrix. So that's where we are getting this negative value from. The more negativities, that means that this amino
More
acid.
mutation.
Yeah, so that.
More negative, more mutation.
No, more negative means that this mutation is not feasible by evolutionary processes.
Favorable by evolution.
So you have to highly penalize it in this scoring system, because ultimately the function of the PAM matrix is you are trying to get, in the process of getting an optimal alignment score, you are trying to exactly consider each matches or each mismatches in a kind of more rigorous way, basically. So for that, the scoring that you are getting, you can get, for example, from this PAM matrix. Yeah.
Thank you, sir. Thank you so much.
Yeah.
So, sir, this data is from literature. Okay. So, as the number of sequences are increasing day by day, so maybe 10 years from now, this data might change.
Yes. So the more number of data that you have available, so the better the quality of the individual data points will be, and the better the quality of your overall matrix will be. So when Margaret Deho, for example, originally constructed the PAM matrix, she had, of course, considered, let's say, the 70 or something group of closely related sequences, right? However, if you see after that, if more kind of sequences are discovered and analyzed in detail, so if you incorporate all of this data as well into this, so this might change again, right? So that is why basically the more advanced sort of matrix instances and so on can be developed. But in any way, PAM matrices are still kind of widely used because it is still reliable. In a lot of sequencing algorithms, you will see that they rely on the scores of these different PAM matrices or Blossom matrices and so on. So Blossom was again developed a little bit later, but yeah. So if you incorporate more available data, if there was data limitation at the point of construction, so when you incorporate more data later, let's say it will become a better sort of representation of that, of the actual kind of values that are supposed to be. Okay. So yeah, the Blossom matrix or the blocks amino acid substitution, it was kind of a different approach from PAM matrix. So it was based on direct observation of every possible amino acid substitution in multiple sequence alignments. Now in PAM matrix, we interpolated, or we, from matrix multiplication, we interpreted the data for the highly divergent sequences. For Blossom, it is exactly from direct observations. So here more than 2,000 conserved amino acid patterns represented by blocks of less than 60 residues are used to create this. And then, for example, a scoring of 62, like Blossom 62, that indicates an average identity of 62%. And again, the overall construction is kind of the same because you have to again convert the scoring to log base 2 and so on. But the difference is that while for PAM matrix, you had to use, if you wanted to compare closely related sequences, if you wanted to do that, you had to use a lower level of PAM matrix. For Blossom matrix, since it is kind of related to the identity, so if you want to basically align closely related sequences, you have to use a higher Blossom number. Whereas in the PAM's case, you had to use a lower PAM number, kind of, right? Again, another sort of Blossom matrix is kind of represented here, and the interpretation is kind of the same here, or similar kind of interpretation. And what I talked about is that for closely related sequences, which of these would be preferable? So of course, PAM lower, that means that is preferable for closely related sequences. Blossom higher, that means that is preferable for closely related sequences. So in this case, for example, the answer could be PAM 40 and Blossom 8 for closely related sequences, right? And then now we can maybe just do a brief about the Needleman-Wunsch algorithm, which can be used to identify the global alignments, right? So what the Needleman-Wunsch algorithm will do is that you have to put both sequences, let's say one at the left hand side and then one at the top, and then first you have to initialize, you have to start from 0 here, and for the horizontal, each horizontal jumps and each vertical jumps, you have to put the gap penalties, and you will just constantly basically accumulate the gap penalties. For example, here -2, after that you have to put the next cell as -4, then -6, -8, and so on. Same, the vertical jumps will be basically the same. The significance is that if you are trying to go from one path to the vertical or horizontal kind of jump when you're trying to trace back, so that will represent an indel or a gap, and that will be needing to be highly penalized. So that is why for the horizontal and vertical jumps, we are accumulating the gap penalties in a way. And what you have to ultimately do is that you have to fill up this matrix, and you have to calculate the score of each of these cells from all 3 cells that you can basically reach this cell to. So for example, here we can reach this middle cell. So this one from 0, from the upper hand -2, from the left hand -2 as well. And when you get these 3 different routes scores, the best score or the most positive score in a way, when you get that, then that will be your score for this particular cell. For example, in this particular cell, if you calculate, you will get 1, right? You have to fill up all of these cells in this way. You have to, for all of these cases, you will have to evaluate these 3 different routes and ultimately put the most positive, or conversely the least negative, you can say, the best score essentially in that. And when you fill up all of this, ultimately the bottom right value in this entire matrix, so that will be your alignment score. Okay. Now, how do you get the optimal alignment? Well, one step that was kind of missing from here is that whenever you put a data point inside a new cell, you have to trace back. So you have to put an arrow to let you know later where this value that you got, where or which cell did it come from. So basically something like this, you can see basically. So this one we calculated this from this 0 value. So that gave us the best score. So we put an arrow here. Then again, this 2 was this arrow. So this 0 was from this 2 arrow, and so on and so forth, right? And what ultimately you have to do is that this bottom right score, which is your alignment score, from that you have to start tracing back until you get to the end, basically, right? So here, if you trace back, you will see that you can get to the initial position by two different paths. For example, this 2 to diagonal 1, and then this vertical, this horizontal 3, then diagonal 2, then diagonal 1, then diagonal 0, this vertical 2, and then this diagonal 1. So this path would be one alignment where the diagonal portions will indicate match or mismatch, and the vertical and horizontal shifts will be indicating gaps being present there, or indels being present there, right? Another path you could have taken, instead of this 2132 path, here you could have taken this 2102 path, and then again proceed forward with the rest, for example 10210. So those two will be giving you different, slightly different alignments. So that means that even if two alignments have the same score, they can ultimately result in slightly different kind of alignment, right? So same optimum alignment score can give you different alignment results in a way. Okay.
So here, so
Yeah.
last bottom right corner 2 is there. So from there, the left corner, slightly left next to
Yeah.
2, bottom
This one.
2. So this one will not go because initially we didn't keep the arrow here. So this 2 is not coming from here. So
No.
that's why we will not consider this 2.
Yes. These two will not be considered here because this two does not lead an arrow to this two, the ultimate alignment score. Okay. So that is why this two is not accounted for. Where this arrow was coming from was basically from this one. So this path you can consider, you have to consider, and then this one got its arrow from this 0 and then this 3. So now this path is kind of diverging here. So either you can consider this 0 1 and proceed forward with this 2 1 0 2 1 and such path, or either you can consider this 3 on the left hand side of 1. So you can proceed forward with 2 1 3 2, this sort of path. Ultimately you will get the same alignment score, and both of these kind of represent the optimal alignment. Of course, in a let's say the actual sequence alignments for a large number of sequences, you will find that maybe sometimes you can get similar alignments, but sometimes also because of the complex scoring systems and such, you can get unique alignments also. Okay, so that's, you have to decide on a kind of case by case basis. But yeah, so you can calculate the value for certain points inside this matrix by just following up the usual process for Needleman-Wunsch. For Smith-Waterman, as we talked about, is that instead of putting basically negative values, you have to just put a 0. Okay, so here again, the process is kind of the same, but instead of putting negative everywhere, you have to put 0, and that will kind of give you sort of the look at where your local kind of alignment can be found. So for example, in this Smith-Waterman matrix, we can see, so in this case, the bottom right corner is not the optimum local alignment score, but whatever is the maximum value inside this matrix, that is the optimum alignment score. And from this maximum value, you will have to trace back the path until you basically get to a 0. So before you get to the 0, until that point, you have to trace back that path. And in this particular case, if you trace back, you will find kind of this sort of pattern. So this GTGA on this side, if you project it, and then this GTGA on the left hand side as well. So those are sort of matching together. So in this case, the alignment will be like this. So here, as I have indicated. So that is the Smith-Waterman approach. Okay. So I think we'll cover or kind of briefly recap the next set of concepts in tomorrow's class. So I hope you have received the email from the NPTEL platform that tomorrow from 6 to 8 we will have another class. I think tomorrow we will also have 4 to 6 class as well. But this will just be an additional class where I'll just try to recap the rest of the concepts that we went over in the different classes. Okay. So for today, I think we'll wrap up the class here. So yeah, so thank you everyone for joining. And again, I will meet in tomorrow's class. Okay. So please try to also attend the other TA sessions as well, the remaining sessions. And then please also try to go through carefully Professor Promiha's lectures, because a lot of the questions that exam you will face, those will be related to the lectures and the similar pattern of the assignment problems that you had solved earlier, basically. Okay. So yeah, so thank you all for joining, and we'll meet in the next class. Thank you.